must keep her through cons and a wishing well. Prayed for a love to call his own, a love to break the spell.
Welcome to the Red Pill Buddhas podcast for red-pilled Buddhas everywhere. Revolutionary, free-thinking spiritual people who've woken up from the mainstream narrative on various levels, and I interview some of the most fascinating ones here. Please also visit thehumanunleashed.com for hundreds of hours of our video content on all areas of health, lifestyle, and much more, and the redpillrevolution.com for our 5-star reviewed book and subsequent publications in the Red Pill series as they come out. Hi everyone, before we go on to the main podcast, Graham and I were chatting, and I got Graham here, Graham Norbury, along as a co-host because he's much more up on this scientific stuff. We thought we'd do a little bit of an intro, or I'd get Graham to do one, about what deuterium actually is, because this wonderful interview we've got coming up with Gábor Somlyai can get a little bit technical at points, but towards the end of it, there's some great practical tips. But for those scientifically minded, you'll be fine. But Graham is the great demystifier, and he can do a great intro here to get you up to speed a bit. So Graham, take it away.
Thank you, Phil. Right, so we're just about to move over to a discussion with Gábor, who is the world's leading expert in deuterium and its importance for human health, human and animal health. So let me just give you a bit of background as to what is deuterium, because it sounds very complex and difficult to get into, but it's actually very simple. So deuterium is a form of hydrogen. Hydrogen is the simplest base atom known to man, which has an atomic mass of 1. Deuterium is just like any other hydrogen atom, except it has an extra neutron attached to it. So its charge is the same, but because it's got this neutron, which is exactly the same size as the nucleus, its mass is double that of a hydrogen atom. And deuterium naturally occurs in the world, and it's naturally present in water. And so instead of the normal H2O, which we all know is 2 hydrogen atoms and 1 oxygen, you can occasionally get D2O, which is 2 deuterium atoms and 1 oxygen. And we discovered deuterium, technically speaking, or officially scientifically speaking, approximately 100 years ago. And you probably know it, if you know it at all, from the phrase heavy water, because water that has lots and lots of deuterium in it, which used to be manufactured in order to use nuclear energy, it was used as a sort of damping fluid in the nuclear process. They began to manufacture that sort of less than 100 years ago. And so they formed heavy water. And what was left, strangely enough, was light water, because they moved more deuterium into their heavy water. And what was left over was this product, which was used to be thrown away, which was called deuterium depleted water. So why am I telling you all this boring stuff? Well, it turns out that the chap you're about to hear from had a very inspiring thought when he was a young scientist in the late 1970s. And he decided that the human body has so many different reactions taking place every second or every millisecond that there must be some simple molecule that was orchestrating this process. And he decided that deuterium and hydrogen might be a candidate. So he looked into it, and he was quite right. And he figured out that the body is very, very sensitive to the proportion of deuterium molecules that make up the body's water compared to the number of hydrogens. As you know, the human body is made up of 70% water molecules by weight. And if you look at, because water molecules are so tiny, the human body is made up of 99.9% water molecules by number of molecules. So basically, every single part of your body is in contact with a water molecule at any given time. And so every single process in your body takes place through the medium of water. And so what Gábor has, what he theorized and then discovered to be true and then went on to test, was that cellular processes are extremely sensitive to tiny changes in the amount of deuterium that you have present. And initially, this was ignored by science since deuterium was discovered up until, so from the early part of the 20th century until the late 70s, when Gábor began to think about this, science, and specifically medicine, just assumed that all water was neutral, and that just because there's a tiny amount of this different molecule in there won't make any difference at all to anything, and so we can just ignore it. So no one looked at it. But he found that if you look at water in the human body, or water anywhere, we have about 150 parts per million of deuterium compared to the total number of million molecules. And that may not seem like very much, and it may seem like an amount that you can ignore. It basically means that 1 out of every 6,600 water molecules is a deuterium water molecule. And so people ignored it, but then Gábor began to look at other things which we do look at in medicine, such as calcium and magnesium. And he found that the amount, I mean, we obsess, some people obsess about the amount of magnesium in their bodies or the amount of calcium. And so that's something that we do pay attention to. And they're present in even tinier quantities. There's something like 6 times as many calcium. The presence of calcium is 6 times as prevalent as deuterium, and magnesium is something like 10 times more prevalent. And so, sorry, I've got the wrong way around, excuse me. Deuterium is about 10 times more prevalent in the human body than magnesium, and about 6 times more prevalent than calcium. And so he decided it was something that we should look at, and he realized that cells pay particular attention to the ratio of deuterium to hydrogen, and they exist in a sweet spot where the deuterium, there's not too much deuterium and not too little. And what he's also discovered is that if there's an excess of deuterium, that tends to be pro-growth, and if there's too little deuterium, or if there's a system that the body has to reduce deuterium. So we have, like in all of nature, you need to have a yin and a yang and a balance. The body has become quite exquisite at balancing these two. So you have to have the sort of the on switch and then the break. And so deuterium levels are used as this braking process in cell growth and many other operations within the cell. So when you start to talk about cell growth and a braking mechanism, if the braking mechanism isn't working properly, you can get into a situation where you have unfettered growth, you have excessive growth, which we commonly know in medicine as cancer, which is terribly frightening, and you can't talk about it on the internet and things like that. But his interest was piqued, and he thought, I wonder if I take some cancer cells in the laboratory and I try to grow them, and I use water which is very low in deuterium, will they grow? And he found out that, amazingly, the cancer cells did not grow when they were in low deuterium water. So he's then spent the last, almost 50 years, 40 something years, studying this and demonstrating that, in fact, if you are able to lower the deuterium in your bodies, you can slow down the growth of cancer, you can reverse the growth of cancer, you can eliminate cancer in many cases, and you can remain cancer-free. You can combine the use of deuterium depleted water with existing cancer therapies. You can have tumours cut out or tumours treated with radiotherapy or chemotherapy. And you can combine that with consumption of deuterium depleted water. And this will dramatically improve the prognosis and the survival time after the intervention. And so these are all amazing feats, which he's spent the last 30 or 40 years documenting. He wrote a book about 20 years ago about how to address cancer and other diseases using deuterium depleted water. That came out in 2000. And he's recently produced another book, which is basically the details of his experience using these techniques in the last 20 years. And so we found that deuterium is something which is overlooked by the vast majority of medicine. It's overlooked by the vast majority of diet gurus and health gurus on the internet because it's so complex. But the underlying theory of it is really relatively simple. The body uses it to regulate growth and metabolism and mitochondrial functioning. And so it's something that you don't need to understand, but you really do need to be aware of. And so we're about to sort of hand you over to quite an in-depth scientific discussion with the absolute world leader in this deuterium knowledge space. If you are interested in improving your health or improving your performance, you should definitely consider this. If you are in any way suffering from what would be referred to as metabolic disease, metabolic syndrome, anything such as sort of diabetes, high cholesterol, all those type of, or obesity, all those type of things, I would strongly suggest that you have a look at his book and have a look at his published papers. And certainly if you are on the road to suffering from cancer or are indeed suffering from cancer, then it's an absolute must, I think, to understand this and consider it as part of a protocol that you would use to try to reverse your condition. I think that should be enough information to generate people's, to pique a little bit of interest from most people. Towards the end of the interview, he does give some practical information. And if you require any more background knowledge, over at our sister site, The Human Unleashed, we have interviewed one of Gábor's students who has worked alongside him during the last 20 or 30 years and has also worked using deuterium depleted water and towards creating drugs. And we also have, I make an attempt to simplify deuterium for anyone who's interested in some of the more complex subcellular processes that deuterium is involved in. But it is a key to health, and it's something we have neglected over the last 100 years as we have changed our diets and introduced more carbohydrates and more seed oils. We've introduced more deuterium into our lives, and this has a detrimental effect on health. And I do believe it's one of the keys to modern day chronic disease. So if you're remotely interested in modern day chronic diseases and what the fundamental basis of them is, this is the interview for you.
That's brilliant, Graham. Thank you very much. And if anybody is watching this who would like to get some links, all Gábor's stuff is below. The links to our Red Pill Revolution stuff and The Human Unleashed will be in the show notes. So that's enough waffling from us, and enjoy the interview. So welcome back, everybody, to my latest episode of the Red Pill Buddhas podcast. And we've got a bit of a scoop today. I had a message from a lady on Instagram called Sylvia, who goes by the moniker of the Yogini next door, saying, would you like to get my dad on your podcast? And it was like, yeah, well, yeah, let's have a look. And then I realized, wow, her dad's Gábor Somlyai. So here's Dr. Gábor Somlyai, and this is great. Gábor is one of the, well, probably maybe even the leading researcher into deuterium depletion, pioneering work. And previously we have we've chatted to Laszlo for our membership site, Laszlo Boros, and he had some wonderful stuff, fascinating stuff to say. And so I've also invited Graham Norbury, my friend and colleague from the Red Pill Revolution team, to come along and ask some more intelligent questions than I can form, because he understands a lot more about this and has a much more scientific mind. But I'm fascinated to see this. For me, you know, in the carnivore world and the carnivore diet of relying very heavily on good animal fats, the healing we've seen is extraordinary. And perhaps a video that I sent to Gábor a few days ago of my mother's breast cancer reversal. I think, you know, this had a lot to do with deuterium depletion, which I didn't understand at the time. But to me, it's fascinating because it intuitively ties in an awful lot of the reasons why carnivore diet and other interventions work. So anyway, welcome, Gábor.
Thank you. It's my pleasure.
It's great to have you on. So perhaps to start, just tell everybody a little bit about your history in deuterium depletion and what you discovered, because you got onto this pretty young, right?
Yeah, so I guess I was a teenager when I recognized that cancer, how important can be, because one of our neighbor had a cancer, and as a young fellow, I saw how bad, how terrible when someone that type of cancer, and as I heard the adults and my parents, I saw that should be a challenge for the humans to solve this problem. And the other good point was when Albert Szent-Györgyi, who is one of the Hungarian Nobel laureate who discovered the vitamin C, had an interview in the early 70s, and I heard a voice, I heard a word, it was submolecular. So he was the scientist who recognized, and he suggested that it cannot be true that the life is so complex and millions or billions of chemical reaction every second, and the fast response to all the challenges should be organized and governed by the huge protein molecules. And he suggested must be something which is very small and very flexible and very fast. And he suggested that the electron should be that submolecular components which finally can be responsible, for example, the cancer, because he also working on how to solve the cancer problem. And the idea came to my mind in 1976 when I said, okay, maybe not the negatively charged electron, but the positively charged hydrogen ion could be that key components in that tumor development. And I make a note. I still have this paper. It was in 1976, May 21st, maybe. And 4 years later, when I was a student on a biology faculty at the university, the idea came to my mind that there is a heavy hydrogen, it's called deuterium. And you cannot explain why, but I was 100% sure that the hydrogen deuterium together should be responsible for the cell division and the regulation of the cell growth. So that's it. That was in 1980. And then I had to wait 10 years. I graduated in 1982. Then I continue my DNA work and molecular biology. I got my PhD. I got a scholarship from the Hungarian Academy of Sciences. I defended my PhD. Then I went to Germany, to Göttingen University for half a year. I came back in 1988. Then we went with my family to the United States. I worked at Columbia, Missouri. I did some gene mapping work. We stayed a year there, and then the year was ended, we came home, and I started to find a job which is related to cancer research. And this is, I go to work to the National Institute of Pathology and the organism. So it was 32 years ago.
That's wonderful. And then when did you start to bring it into sort of your profession and set up all these companies and you know, that you've done, and to cover all of this this research that you've done and bringing it into production?
It was so the very first data in the how did I start it? So the simplest way is to check. So the question was whether the nature occurring deuterium has any role in living organisms. And the point is that everybody knows that deuterium is present. It's almost 100 years ago it was discovered. Everybody knows that the deuterium behave different chemical reaction, and everybody was aware that the ratio between the hydrogen and deuterium is 6,600 to 1. And that was the reason that was ignored, because the people may believe that, okay, it's only 6,600 hydrogen, there is 1 deuterium. Why should be any role of the deuterium in the living organism? But if we investigate the deconcentration in the concentration of millimole, it turns out that the deconcentration is 30 millimole in our body, but the calcium only 2.5 millimole, the magnesium only 1 millimole. So my question was, what will happen if we reduce that pretty relevant deuterium concentration in the tissue culture or in any living test system? So in my very first experiment at the institute, I investigate whether we can modify the cell growth if we prepare media with a low deconcentration. And the finding was that we could reduce the growth rate of the cells when the cells were growing in the media in a 25 ppm deconcentration. And the next experiment, we transplanted human breast cancer to mice, and we changed the normal water for deuterium depleted water, and 60% of these mice were healthy at the end of the 3-month study. Still 99% of the mice perished. They consumed normal deuterium depleted water. And we published our very first paper in 1993 in the Phelps letters. And the basis that we established the company based upon that discovery and based upon that data which showed that the deuterium has a very important role in the living organism, and the shortage of deuterium is a big challenge for the cancer cells. And I hope that way we can develop some kind of drug which modify the DH ratio in the body, and that way we can selectively kill the cancer cells. So this is how we started 30 years ago. The company will be 30 years old next year.
So Gábor, just to clarify in my mind and also for the listeners, I understand the aspects of hydrogen protons flowing through the mitochondria, and I understand that the mitochondria seeks it doesn't want deuterium within the mitochondria. And then I've heard you discuss the sodium hydrogen on the cell membrane, the port, and from if my understanding is correct, the cell itself prefers to eject hydrogen and take up sodium, and that itself would increase the ratio of deuterium in the cell, and that is a growth signal. Is my understanding correct?
Yeah, and this is what we wrote in the very first paper. So putting together the pieces, what I have read, and independently from me, that when a growth hormone binds to the membrane, that will stimulate the sodium hydrogen, that will eliminate the hydrogen and pick up 1 sodium, and that causes a pH shift. And everybody agreed that this is somehow mediate a signal for the cell to multiply. But much more important, that there was other paper published in the Nature. They isolated the ATPase gene from the yeast, and that gene was used to transform the animal cell line. And they could prove that the ATPase enzyme were present in the membrane, eliminating the hydrogen, and everybody was surprised that caused tumor in nude mice. So not 100 different cancer genes, not 100 different mutation, one extra ATPase gene, which selectively eliminate the hydrogen, could cause tumor in nude mice and cause tumorous phenotype in vitro. So, and this is what we published in the paper, that when the growth hormone binds to the membrane, this is what you said, and activate the sodium hydrogen, that will discriminate, and selectively eliminating the hydrogen, it means that the DH ratio start to increase in the cell. And I said, and we said in the paper, not the pH shift is the key signal. The increasing DH ratio is the key signal for the cell. And the other point is that just recently we published a paper in the cancer control. We investigated about the expression of 700 genes. It's over 200 was a cancer-related gene, and other 500 is kinase gene. And we tested the gene expression in deuterium-depleted media, in normal media, and deuterium-enriched media. And the point was that hundreds of gene expressions were modified on a higher D concentration. So when any membrane transport process preferring the hydrogen, and that way increasing the DH shift, that simultaneously will be able to switch on or switch off genes in the total genome. And the point, so when I saw that people are focusing on the mitochondria and the membrane. So there are two processes in the cell. The membrane transport is able to increase the DH ratio. The mitochondria is going to keep on a low level the DH ratio, synthesizing and producing deuterium-depleted metabolic water. And these are imbalanced in a healthy cells. And if we go back, and what is the only common feature in cancer cells, it is the damaged mitochondria. So the reason that the cancer cells can very rapidly and so frequently trigger the cell multiplication, the cell division, because no healthy mitochondria, no deuterium-depleted water production in the cell, and this is no break in the system. And so the deuterium depletion and the DH ratio is not just relating to the mitochondria, it is relating to all the metabolism, it is relating to all the gene expression, and that will govern the movement and the regulation, all pieces, all part of the cell. Mm-hmm. And in a healthy cell, they are balanced. The damaged mitochondria allow the cancer cells very easily lift up the DH ratio to the threshold.
And so in a healthy cellular environment, we have this natural balance where the membrane begins to lose, the membrane releases hydrogens and keeps deuterium.
Yes. And
the mitochondria continues to produce lower deuterium water. And that's a sort of like a cycle. It's like everything in nature has to have a yin and a yang. And so if that's kept in perfect balance, you go from one end and then there's a sort of break, and then the other end, and then it comes backwards and forwards. And if it stays like that, you remain cancer-free and everything is fine.
Yeah.
Could you say a little bit about what is the cell in a healthy cell? How does the sort of growth stimulate? Let's take a child which is growing and he's having to develop. I presume at that point the cell is trying to grow, and so it's going to be increasing. It's going to be trying to increase its deuterium content by getting rid of hydrogens in order to stimulate healthy natural growth. Is that correct?
So far, first we have tested the side effect and toxic effect of deuterium-depleted water in my study in tissue culture with dogs and cats, and even in the phase 2 clinical trial what we have conducted, and so far we didn't see that the consumption of DDW would cause a blood counts problem or vomiting or hair or loss of hair, whatever. So I don't know how fast we should reduce the deconcentration and how low we should reduce to cause any toxic effect or any side effect. The reason is that we didn't find, because immediately when we modify the DH ratio, someone start to consume deuterium depleted water, that will challenge not only the cancer cells, but all the cells. They have to somehow adapt to the lowering deconcentration. And this is a big difference between the cancer cells and the healthy cells, that because the metabolism is normal in the healthy cells, they will handle that the deconcentration is getting lower and lower. And we must be sure that the cancer cells also working on to adapt to the lowering deconcentration. And they are able to adapt to the lowering deconcentration. So immediately when we modify the deconcentration, all the system will react, and the healthy cells can react. We have a couple of children who had cancer, and they grew up, even they were consuming deuterium depleted water, so we didn't see any kind of problem with that. But so the real challenge that the ability of the cancer cells is present, and of course they will working and the metabolism is going to handle it. And if someone got huge cancer, I mean 10 centimeter in liver metastasis or whatever, we cannot say that there will be cured within overnight, because it's a huge mass of cancer, and the cancers will adapt to the lowering deconcentration. And this is another challenge, the dosage of the depletion. So the protocol, what we suggest now, that we have to start one type of deuterium depleted water. At the beginning, it goes 1 ppm decrease per day, and after 30 days, it's getting smaller, and after there will be an equilibrium. And after 2-3 months, we recommend to change for a lower deconcentration, because that way we can again challenging the cancer cells and all the cells. And this is a big game between the healthy cells and between us and the tumor, to challenging as much changes in the deconcentration, which trigger the apoptosis, which cause the redox balance becomes disbalanced, increasing the radicals, and that radicals will trigger the apoptosis. So it was typical, for example, when sometimes we couldn't see the efficacy of the DDW, and spending lots of time with people just talking and speaking and listening them, it turned out that those who were taking lots of antioxidant beside the consumption of DDW, in that case, that prevent the efficacy of deuterium depleted water, because we need the radicals. And
you need the radicals to signal the apoptosis.
Yeah,
right.
Yeah. And we published a paper with the Vanderbilt University when we checked the anti-aging effect of the mice study, and we could prove that immediately the superoxide dismutase expression increased when we use the deuterium depleted water. It means that the healthy cells recognize the lowering deconcentration. It may cause increasing the radicals, but the system immediately respond and try to eliminate the radicals from the system. The question is whether we can trigger so much radicals in the cancer cells to trigger the apoptosis, and that way we can cause tumor regression.
Right. Okay. And so here, what we're trying to achieve is, we recognize that cancer and metabolic disorder have their root in mitochondrial dysfunction.
Yeah.
And mitochondrial dysfunction in the main seems to be driven by excess deuterium within the mitochondria, or let's say an inability to deplete deuterium.
So what we can say that any chemical reaction, the hydrogen is preferred, because it's much easier to split the bond and so on. also cited a paper in a very first paper that it was proved that the hydrogen ATPase does not accept deuterium as a substrate, which means must be a very strong discrimination in that enzyme and preferring the hydrogen to eliminate. But so the question is, what is the cause and what is the effect? And the deconcentration will be determined what we drink and what we eat. And that will give us an average deconcentration. And I think that the big changes in the last 60, 70 years, when the fat was said, this is a bad thing, don't eat fat, and the carbohydrate was preferred, and they were said, okay, eat more carbohydrate, cover the calorie intake 60, 70% with carbohydrate. And because we check the deconcentration of different food, and the carbohydrate will produce 150 ppm deconcentration, the pig fat 118 ppm deconcentration. So as the society and the population is starting to eat much more carbohydrate, then the average deconcentration is increased. And we eventually checked the deconcentration of the blood, 30 people in the human clinical study. We found that 145, 150 ppm, that was the deconcentration. So very close to the 150 ppm. And I guess my grandfather, if we could measure it, his blood deconcentration, I guess, should be about 130 ppm, because he ate the fat and the bacon and so on. Mm-hmm. So based upon that, the average deconcentration is much higher now. That can modify the gene expression, that can modify the enzyme activity, and that can lead to the mitochondria is getting damaged. That may increase the mutation. And altogether, this is the reason we say that there are so many cancer, there are so 200, 300 million diabetic patients, high blood pressure, and all these things, because we are living our lives on a higher average deconcentration. And if we can intervene, and the ketogenic diet, which reduce the deconcentration, we go back to the natural level.
Okay. Can I jump a quick one in there, Graham? Gábor, when we spoke to Laszlo last time, he said something very interesting. I mean, we all know that the seed oils that they make are incredibly toxic, and people see it as a sort of omega-6s and trans fats and whatever. But he said something very interesting, that when the body gets an overload of deuterium via carbohydrates and stuff like that, it's bad enough. But when it comes in via seed oils, it's far more difficult for the body to get rid of that. Is that something that you might have seen or thought about? Because I think it just shows that it's just another way that the corporations and the ridiculous advice we've had is harming us to get onto these things and saying they're healthy, and animal fats aren't.
I was going to ask the same question. I think what Laszlo said was that if you eat carbohydrates, we have the Krebs cycle, which has the ability to strip out the deuterium so it doesn't make it into the mitochondria. But he said that if the fat, if energy from fat comes into the mitochondria, there's no control mechanism built in naturally, because we're supposed to be eating natural fats which are low in deuterium. And if you have industrially produced seed oils, they may be high in deuterium, and if they are, that deuterium just goes straight into the mitochondria and causes the damage. Have we understood that correctly?
So yeah, so what we have measured these basic food components, but it's sure when we artificially modify the food, that can modify the DH ratio in a given molecules or a given food. And that can very significant effect, that can cause very significant effect on a living organism. So yes, it can be one reason that we don't know when for example, they hydrodize and they put the hydrogen to different oils and whatever. We don't know the ratio of the hydrogen gas which is used up, and that way they modify the DH ratio. But one more important thing, that we had a couple of international congress, and I invited Richard Robbins for the second one, and he is a person who could measure the deconcentration on a linolenic acid, and there is 18 or 16 carbon in that molecules, and he could measure, for example, that the carbon 9 and the carbon 13 deconcentration is only 60 ppm, and left and right is higher, which says that how sophisticated the molecular system, the fat synthesis, that the deuterium not just randomly are present in a fat molecules. It is dedicated depending on the number of the carbon. And what we say, what I say with this deuterium hydrogen ratio, that what we should address, how complex a single cell. There are 2,000 biochemical processes in one single cell, and there is a 1.8 meter length DNA in a nucleus, which is only 10 micrometer in diameter, and each molecule is working perfectly. And these are not organized on a molecular level, that these are organized at that submolecular level. So changing any part of the metabolism, the DH ratio will interact and will modify other part of the process in the cell. And that can, as I mentioned, the gene expression and all the other things. So the point is that should be the basic regulatory system. We are working on a molecular level, and the molecular biology is awesome. So we know everything. We know all the molecules. We know the structure of proteins, but they are governed from the submolecular level, just changing the DH ratio, and the membrane transport and the mitochondria has a key role in that process. So whenever we send molecules by the food or water, and the deconcentration of any kind of food or the liquid will determine and will influence all the process in the cell.
So if you were to sort of make a suggestion, if you had somebody who was perhaps suffering with metabolic disorder, which we could argue is a preliminary pre-cancerous stage, because if they ignore that for a length of time, it could develop into cancer. So you'd have a situation there where the mitochondria were not functioning well. The energy, the electron flow was slowed. Therefore, all the field, the electric and magnetic fields which control the flow of all the particles would be lowered, and you would have lower energy produced. So your fundamental theory is, if we lower the deuterium within the mitochondria, we enable the cell itself to bring itself back into balance.
No. My concept is not. I don't believe that with the lower deconcentration we can repair the mitochondria. The concept is to kill these cells. So those cells which are not able to adapt and handle the lowering deconcentration, they should trigger the apoptosis.
Okay.
I don't believe that we can repair the mitochondria. We have to select all the cells which are not healthy. And the new generation, the cells which survive that stress, they will be healthy.
So you're allowing the cells to, you're stressing the cells and hoping that they will trigger apoptosis due to free radical signaling, which returns to normal.
Yeah, this is what we have published, several times. Yes.
Okay.
And so when someone got a cancer, and the size is decreasing, it must be because there is a necrosis, there is a apoptosis triggered by deuterium depletion. And so we see it macroscopically as a tumor is shrinking and the size is decreasing.
Could I pass something quickly there? Is this perhaps a similar mechanism by which people who are getting good success on dry fasting? There seems to be some research coming out from, well, it's quite old research, I think now, from Russia, and people are trying this where they say an 11-day dry fast with absolutely no water, no nothing. And it sends the body into that similar kind of state of sort of increased apoptosis and autophagy. And also some of the people who are talking about the dry fasting are also saying it helps with the cell producing sort of deuterium depleted water within itself. What do you think about that one?
Yeah, so this is what we use out. So when there is a radiotherapy, we hope that the cancer cells are more sensitive for the radiation and they die. When we are applying any chemotherapeutic agent, we hope that the cancer cells will be more sensitive for that drug. Changing using high concentration vitamin C, we modify the environment, we modify the parameters, and we hope that the healthy cells can adapt faster and can handle it more adequate way than the cancer. So changing any parameters is always a challenge for all kinds of cells, and because the damaged mitochondria, because the damaged metabolism, the cancer cells are more sensitive, and this is how we try to be very specific and killing only the cancer cells. Yeah, it's the same system.
And the concept of damaged mitochondria, we've heard discussions of if deuterium is used instead of hydrogen within the mitochondria. You already mentioned that the ATPase doesn't like deuterium, and the sort of conceptual visual model is of a rotor which accepts hydrogen protons and spins, and the deuterium, because it's double the mass, doesn't. It kind of clogs up the motor, as a gross way of saying it. Are you happy with that explanation of damage due to deuterium getting into the rotor? Does that mean that if every now and again too much deuterium gets into the rotor, it damages it, therefore that mitochondria, that particular chain within a mitochondria with the ATPase is damaged and needs to be repaired, and therefore the cell's energy drops because and if it goes on, you get more damaged rotors and there's less energy produced. Are you happy with that concept?
It should be, but we should see it in the practice, and we should prove it in an experiment. But maybe our experiment proved that this is true, because we have run a study with athletes. So we made a loading test with athletes and tested the blood parameters. They consumed one of the loading test. And what we found that before they started to consume the deconcentration, after the loading, the blood sugar level first start to decrease and then start to increase because they need the energy. And the lactic acid appeared in the blood. After they consumed the DW for 44 days, we didn't see the increase, and it's much later the lactic acid, which means that the mitochondria worked much more properly on a lower deconcentration. They could utilize much faster and much better the glucose, and that should be one way that really the mitochondria prefer and work much properly on 130, 125 ppm. But it also can be that, for example, I guess maybe the deconcentration can modify and influence the division and the mitochondria number within the cell. Because maybe in that athletes, we could see a better data and the lactic acid occurred later, because maybe the mitochondria number increased within the cell. Maybe the lower decontent stimulated the multiplication of mitochondria. I don't know. So, and can be another 100 thing which can modify that we could recognize that the lactic acid appeared later in the loading test after consuming DDW. So I don't know the right answer. The deuterium and then this bigger mass could be one reason that the system can be very sensitive for the double mass deuterium.
Gábor, could I ask you about I think you mentioned at something I was listening to of yours, that there are some types of cancer that respond better than others, or is it more to do with the stage of cancer? Well, I mean, I see the same thing where a lot of these results of these interventions are kind of confounded, because I think when they were treating a lot of the experiments of sort of trying to treat cancer with keto diets, they were constructing it with sort of really crappy oils, and then it wasn't working. But as soon as they started using the animal products, then they started to have far better results. It's not, I don't think, just a matter of ketosis. Obviously, there's something else going on there. But is there a certain type of cancer that this is more difficult to get to respond?
Sure. We have seen about 50, 60 different cancer types, and we say that street cancer is very difficult. It's a melanoma, pancreatic cancer, and glioblastoma. We consider that these three cancer types, we suggest a different protocol, we suggest a different mode of the application of DDW. And of course, so it would be very strange if all the cancer would respond on the same way, because each of them has a different molecular background, a different structure, and so on. But the good news is most of the cancer responds quite well. And even I say that the glioblastoma is a very tough type of cancer. Based upon our data, we can get a 3-fold increase in median survival time, because in our case is 45 months, the median survival time. And this is the reason I say that we are at the beginning of something, and we have to work on to figure out all the details of this mechanism and to find other molecules combining with DW to even increase the efficacy of deuterium depletion. We have seen lots of good combination with the existing drug, Herceptin, Sutent, Glivec. These are very, very good combination with the deuterium depleted water, because they are good drugs, because they are also modifying the DH ratio. And those drugs combining with DW, we can get a very strong synergistic effect. So this is what we have to continue and finally to have a very, very safe and very effective drug.
Cool. Have you heard, if you just quick one there, Graham, but just wondered on that, have you heard of somebody called Andrew Scarborough? No. He's great. He's a friend of ours, and he did have surgery for his glioblastoma, but then he's been controlling it, and he's been fine for a long, long time with a carnivore diet and deuterium depleted water. I think there's quite a few interviews with him out there. As a young guy, he's many years on from having the surgery and controlling it with that. There's no recurrence of it. It's nice one. Yeah, Andrew Scarborough is cool. Sorry, carry on, Graham.
No, what I was going to say was, so Gábor, what you're presenting after your many decades now of work is that a completely safe product, which is deuterium depleted water, if you consume deuterium depleted water, let's say that 105, 120 parts per million for a few months, if you're a middle-aged person who's aging slightly, you don't have cancer, but you may be borderline metabolic disorder, or maybe you're an athlete and you want to improve your performance, simply consuming deuterium depleted water as your drinking water for 2 or 3 months is enough to allow your body to rid itself of the poorly performing cells, and your overall health will improve, and there is no contraindications to following this protocol. So you have a perfectly safe, what almost seems to be a miracle product, but it's based on really deep, proper science. And so, although it's not the cheapest water in the world, because it's specially made, it is something which people are willing to spend hundreds of dollars or thousands of dollars on their health in terms of supplements and treatments. But this is something which is relatively simple and absolutely safe, that anybody can use. Is that the case?
Yeah. So do you know, I have spent so many years, and this is the reason I published the second book. And I in the book there are several protocols, depending on the cancer type, depending on whether prevention, or depending for the athletes to improve your physical activity. And on a long term, I would suggest for healthy people to reduce the incidence of cancer over 35, 40 age, should consume every year, every second or third year for 3-4 months deuterium depleted water. That should be 125 or 105. That would cause a decrease in the deconcentration, and that would be enough to reduce the incidence of cancer. And why can I say that? Because we published in a cancer control a paper. In that paper, we evaluated 204 cancer patients. They were diagnosed, they were operated, they were tumor-free, but they were consuming deuterium depleted water in spite that they were tumor-free. And the cumulative follow-up time of these 204 patients was 1,024 years. So average 5 years we could follow each people. And we lost 11 patients out of 204 patients during the over 1,000 years cumulative follow-up time. And out of 11 who died, 8 of them died after 2, 3, 4, 5 years, they stopped drinking DDW. So those who were diagnosed, consumed DDW, had a break, then consumed again, then had a longer break and consumed again, they stay tumor-free. Those who consumed and then they stopped drinking, after a couple of years, the tumor appeared, and unfortunately, we lost them. So that should be a good example that everybody has cancer cells, everybody is controlling the immune system and all the thing. There is a stress, there is a high dose of something, chemicals, UV or whatever, and the cancer has a chance to grow. After 4 or 5 years later, we will recognize the tumor. But when there is a treatment, there is a shift in the deconcentration, that should be enough to eliminate this small group of cancer cells, and the body will be again in a healthy, balanced situation.
So I think that's what's so fantastic about this, about your discovery and about the water itself. And we have millions of people throughout the world who are worried about their health, and they're concerned enough to go to see specialists, to go fly around the world, to go and have special treatments. Whereas what you're saying is this simple procedure, which is so simple, it's you replace your drinking water with deuterium depleted water for a period of 3 or 4 months, and you do that every couple of years. That is enough to clear out the damage in your system and maintain, or indeed improve, your health.
This is what I say.
And that's the most fantastic thing ever, isn't it? And you might say, well, why on earth is this knowledge not more widely known and more widely used? Is it down to the profitability of big pharma, in that they would rather sell medicines and they don't?
No, I don't believe so. No, I don't believe so. So I think to change the paradigm is not easy. So I say something which is very strange. Why should anybody accept that I say, okay, deuterium depleted water would be good to treat and prevent or cure cancer. So we had to accumulate the data, and this is what we need to the breakthrough. And of course, we need the pharma industry who should use up that type of knowledge, that type of data, what we say that when the tyrosine kinase was identified, it was in 1982 by Bishop. Everybody was happy. We have to find the tyrosine kinase inhibitors, and that way we will be able to treat the cancer. So, and that helped a lot. And we have a couple of good drugs which is targeting the tyrosine kinase. Now, what I say, here is a new regulatory system, a submolecular regulatory system. We have to intervene into that level to the living organism, and that will help us to find the answer and to have a good and safe and effective drugs. So that's it. And I'm looking for the cooperation with the pharma industry for 20 years. They are very satisfied with the data, but it's very hard to break through that. Anyway, we have to move in that direction, and we have to combine the existing therapy with this deuterium depletion to have those data and to have the best chance to treat the people.
Gábor, have you looked into you know, there's this thing about the blue zones, and all the vegans are saying, you know, oh, the blue zones, and it's because they ate beautiful plant-based diets, which they didn't. Their diets have been horribly misrepresented over the years, and they ate, you know, like in Okinawa, where they ate a hell of a lot of pork and lots of fat, and they pretend that they're plant-based. But have you looked into any of the sort of areas, or has anybody as to whether, I mean, there's a lot of factors that go into it, great lifestyle, being outside, being in the sun, and all of that kind of thing. But is there any kind of correlation that you or anybody has seen between sort of areas of low deuterium water and longevity and very healthy populations?
So I was in the Caucasian, and I brought some water. It was below 140 ppm, and the people are very healthy over there. In Grönland, they eat only fat, the people in Grönland, and they didn't know the cancer very long time. I guess there is very strong correlation, and there is one way. Do you know one way of the explaining and so why, what is the cause of cancer is always so one school is said that because the mutation, that there is some genetic modification, this is the reason of the cancer. And what should be the connection between the deuterium and the mutation rate? So we published again a paper in the PLOS1 when I asked the group to check whether deuterium depleted water reduced the mutation rate in the bacteria. And what we could publish that 9 out of 15 experiments, the 2 ppm water reduced significantly the mutation rate. And if we think about that, in our body, there are 10 to 16 cell division during all of lifetime, 10 to 16. And think about that, each genetic program is 3.2 billion base pair. So we are synthesizing trillions of kilometer DNA. And if a couple of % we can reduce or increase the mutation rate, that again can lead to a higher risk of cancer, or maybe we can reduce the risk of cancer. And I say that when we are living our life at 150 ppm, it's a chance to have a mutation rate. When we go to the 120 ppm, so slightly on a lower deconcentration, that reduces the mutation rate, which can again help us to get cancer in an older and a late stage of our lifespan. And all these things depends on the DH ratio. And maybe in that region, in Grönland, in Caucasian, or wherever, if there is some 10, 15 ppm lower deconcentration during a lifetime, it counts. It can be very important.
Slightly separate question. Are you familiar with the work of Gerald Pollock and the fourth phase of water.
Yeah, yeah.
I've always thought that it would be interesting because he's looking at hydrogen atoms and their movement and their separation within water. And as far as I'm aware, he does not consider deuterium concentration in his experiments, and he's used light, he's used different wavelengths of light to measure the different effects, and he talks about the viscosity of the water. And we know that light is present in mitochondria, and maybe light is what controls these thousands of subatomic processes. It would be very interesting for him to redo his experiments using different concentrations of deuterium within the water. Is that something that you've considered?
Yeah. So when we have an ice, and in the ice the water molecules are in a fixed position, and it means that all the hydrogen is moving simultaneously with the other, one moving into that direction, the other direction, and they are harmonized. If we drop one deuterium into that system, that will disrupt 100 connection within the ice. So it has so big influence on deuterium because the vibrational frequency is so different comparing to the hydrogen-oxygen vibrational frequency that can disrupt. So in the experiment, what he used, I think he need a pure H2O, because the pure H2O will keep that structure what he recognized and we suggest. If there is a deuterium, that can disrupt this zone of the water, and of course it has a very big influence of this whole system. Yeah, it's I guess it's very important. We also know that, for example, there are several proteins that require a hydrogen to be bound to the protein to work properly, and one D bond can modify the efficacy one protein structure. And it has been shown that if you deuterate the water, the protein will be more solid and more rigid because we replace the hydrogen for deuterium.
Because I'm interested in the concept that within our mitochondria we make use of this exclusion zone of water, which he's referred to. And it seems the greater the exclusion zone, the more efficient the mitochondria seems to be working. And so I'd be very interested to see Pollock do his experiments with extremely low deuterium water. And my guess would be that he would achieve a higher exclusion zone.
Yeah, I guess so. Yeah, you're right. Yeah.
So, Gábor, one thing I was wondering, a lot of people I've heard saying that sources of deuterium depleted water. I guess a lot of people now are going to be, who've been listening to this, are going to say, hey, I've got to try this. I've got to have a go at this. But I hear that some of the sources of deuterium depleted water are not what they say they are. And what should people look out for? And which do you think? Where's the best place to get it? We get it from you?
Of course, I can offer ourselves because we have it. But I think so. So to produce deuterium depleted water is not a secret. It requires some knowledge, but it's easy to do. I guess more important how to use it. And this is very important that we are going to register a drug because someone got cancer, we need to diagnose it, and we need to treat it, and we need to follow it. So in the long term, I guess it's not the way that someone feels something in the breast and go to the supermarket and start to consume some kind of water. So this is one reason I suggest on a long term that two types of products should be on the market. One which would be very close to the natural level. Healthy people should consume it just to reduce the incidence of cancer, to feel better. And the lower D concentration should be a drug, register it, dosage, and following the patients. right now we are in that middle of this story. People can buy it. And again, I hope the book contains the main rules, how to use it. I try to collect all the knowledge and share it with everybody, because I didn't believe that it should take 30 years to be only here and not having a registered drug. But we are still working on it, and we are waiting for investors. We are waiting for the pharma company to join and to move forward and to get good drugs on the market. So people, I guess, look around, check it out, what is the deconcentration, and check it, how to use it, what is the best way to apply it. This is a key issue, I guess.
And your water is it's available, I understand, in the United States. Yeah. Is it how broadly distributed globally is your water?
We have exported 50 different countries. We have a couple of countries where we have a distributor from China, United States, UK, Netherlands, Australia. And these are food products, so we do not claim it. We talk about the science. And independently from me, I guess more and more company will appear and will sell deuterium depleted water. And this is the reason we must move forward to have a drug, that by the time it will be accepted, then we need a drug and everybody will get in a proper way the drug.
So the water, as you said, the water, which is your current product, which is considered as a foodstuff, that's freely available for anybody to buy.
Yes,
it's absolutely safe to use because it's really just water. And if people purchase your new book, your old book, which is now 20 years old, I guess, that was a fantastic book. So I'm very much looking forward to reading the new one. And I would imagine, given the amount of detail and experience that went into the first one, with an additional 20 years of knowledge with the new book, I should imagine that anybody with an interest in improving their health, or anybody who is perhaps faced with the threats of cancer, would be well advised to buy that book and consider purchasing the water. I'm not pushing your product here. It's the science here is so clear. The safety aspect is absolutely unquestioned. That there is no danger to consuming this product. And I think everybody who is in a position of wanting to improve their health for whatever reason should seriously consider doing this, even though it sounds like a strange concept, but it really is so fundamental that it's been overlooked by the vast majority of medicine. And it's crazy that it's not more widely discussed, because it's so obviously beneficial for perfectly explicable reasons. I think it's just the simplicity of it is fantastic.
The nature is not so complicated. So there are very simple rules in the nature. If we find them, we can be happy and we can use out. So we must follow the rules of the nature. We cannot change it. We just discover it if we are very lucky. So this is the reason. You are right. We should do that.
And do you think it's fair to say that deuterium accumulation, or as we age, we lose, or put it another way, that aging is really a measure of deuterium accumulation in the wrong places?
Yeah, that was proved not only with the deuterium, but other heavy isotopes. So oxygen 18, nitrogen, it was proved. I remember 40 years ago when I started that, I saw that type of literature that with the aging, the heavy isotopes concentration is increasing, because they are never preferred in the chemical reaction. And and that correlation exists.
Gábor, this is something that somebody always, quite a lot of people ask me, is when you're buying deuterium depleted water, would you, say you're shipping it a long way, Is it worth buying the very, very low concentrate and then diluting it yourself? Or is there some advantage to getting the exact level that you want in the bottle originally?
It depends on the shipping cost, because the production of the water, the cost is exponentially increasing as we are going to prepare lower and lower the concentration. So reducing from 150 to 100 ppm, 50 ppm, let's say 1 unit, from 100 to 50, it should cost 10 units. So the price goes up exponentially, but it depends on the shipping cost. The best way, if the people can buy those concentration, what they are going to consume, but sometimes because the shipping cost is too high, maybe cheaper to buy the 25 ppm and dilute back to 105 or whatever.
Is it still the case that the most of the, if not all of the production facilities of deuterium depleted water are in Russia, the Eastern European countries, and maybe China, because of their history with using heavy water for nuclear weapons? Is that
so? Those facilities which are able to produce heavy water are able to produce deuterium depleted water.
Yeah.
That never happened in the same time. But in Hungary, we have a GMP facility. We set up a facility just to produce DW for the clinical trials. So our company itself has a GMP facility, but you are right. So those countries used to the nuclear power plant used heavy water, and that required a facility to produce heavy water. Recently, the nuclear power plant don't use heavy water, so these big companies, they were closed or they were dismantled. But this is the reason that Russia, China, and all these countries has that type of facility.
Are there any producers of this kind of deuterium depleted water in America or in sort of the UK or Germany or anywhere like that?
I don't know that. No, I don't know. We are planning to set up facility in the United States, and because the best way to produce the DDW where we are going to marketize or distribute. So you are right. So from bringing water from Hungary to China or Japan or whatever is not an easy way. On a long term, that should be the strategy to set up facilities different part of the world.
I don't think I have any more questions.
No, me neither. That's fantastic, Gábor. Thank you so much. I think that's really fascinating. There's something for the scientifically minded and for the scientifically retarded like me. And I think that's very, very cool. Thank you. Thank you, Graham, so much for helping out. So Gábor, we'll put some links to all your work and books and whatever in the show notes, but tell people where they can find you anyway, and where they could discover about this, get some water and whatever. So we are in Hungary. Our water is called Preventa. People can order it on a website, preventa.hu. We are ready to discuss and communicate with the people who are interested and they would like to know the details, whatever. We have distributors, I mentioned, they can find it on the website in different countries where they can directly buy the product. But the most important, not the water, the knowledge behind that. So this is the reason I worked on 3 years to put together this book and try to gather all the knowledge which can be very important to share with the audience and healthy and cancer patients as well. Marvelous. I'm looking forward to reading it too. So yeah, everyone grab Gábor's book, and thank you so much, Gábor. Thank you very much. Thank you. Thank you.
Thank you.
Thank
Excellent.
you.
Thank you very much.
must keep her